Hello students, today we are going to study about linkage and recombination from the chapter Principles of Inheritance and Variation. So, before going to this topic, just explore this picture. Can you tell me what you see in this picture? Well, here you can see a yellow color circle intersect with the blue color circle, or in other words, we can say a few part of this yellow circle is passed to this blue color circle, and few part of this blue color circle is passed to this yellow part, and in the middle you can see the collectively produce a different color, that is green. This is what we observe in this picture, isn't it? Now you can see there is 3 different pictures in this total whole slide. So what is the difference, or what differences do you see in these pictures? Observe these 3 pictures and just try to tell what you exactly see in these 3 pictures. Well, in this first picture, try to observe there are 2 different magnets placed distantly, isn't it? In the second picture, the 2 magnets are placed little closer than the first one, and in this third picture, the 2 magnets are very close that both the magnets is attached to each other. Or we can say they are very much linked with comparison to other 2 cases. This is what we observe in this whole picture. So, what we see in the 2 previous picture, if I try to summarize those things, then the observation what we saw in the previous 2 picture is when 2 things cross each other, one half is passed to the other, and when 2 things are placed close to each other, they are more linked. Now look carefully these 3 pictures. In this whole picture you can see there is 3 cases. Just like the color circles exchange their parts, here also we observe the same thing. Here you can observe 3 different cases where 4 different kinds of gene present. So basically 4 different kinds of genes are present on 2 homologous chromosomes, that is capital A and capital B, and in this chromosome you can see small a and small b. In the first picture you can see capital A and capital B is very far from each other on the chromosome, and the small a and small b is also far from each other. So, when crossing over will take place between this chromosome, the chromosomes will exchange their segments. That is, this small b will move to this part and this capital B will move to this part, just like we saw in the colored circles. Now observe case 2, when gene A and gene B, that is capital A and capital B, in this case you can observe they are little closer than this case. Similarly, you can observe small a and small b are also little closer than this case. But here also we can observe the crossing over takes place even if the genes are present little closer. Still, the crossing over takes place. That means even if the genes are closer, if crossing over takes place in this part, both the genes will be separated and transferred to other part, like the capital B will be transferred to this part and the small b will be transferred to this part. But can you see this case 3? Here you can see the capital A and capital B gene and the small a and small b gene are present almost in contact, or we can say they are very tightly linked. You can see they are almost present in contact with each other. So, do you think there is a chance of crossing over? Or can I ask that what will be the probability or chances of crossing over in this third case? Well, it is nil. That is, the crossing over is not possible in this case. Since we talked about crossing over, that crossing over generally results in the exchange between non-sister chromatids, which results in some variation. So let's see, students, what actually results from such crossing over.
Crossing over during meiosis allows recombination of genes between homologous chromosomes. This alters the linkage between genes on the same chromosome. Crossing over occurs frequently during prophase 1 of meiosis, when homologous chromosomes associate with each other in a structure called a tetrad. First, consider the possible inheritance of alleles for 2 genes on a particular chromosome when crossing over does not occur. Recall that one member of each pair of homologous chromosomes came from one parent. The other member came from the other parent. In this example, one parental chromosome had alleles capital A and capital B, while the other had alleles lowercase a and lowercase b. Without crossing over, each haploid cell produced will inherit a chromosome with the same combination of alleles as was on one of the parental chromosomes. Thus, the arrangement of linked genes is not altered in this case. Now consider the case where there is crossing over in the region between the 2 genes. Since the crossing over is only between 2 individual chromatids, the possible haploid cells after meiosis have 4 different genetic combinations. 2 will come from the chromatids that did not participate in crossing over and thus will still have the parental combination of alleles, either capital A and capital B, or lowercase a and lowercase b. The other 2 will be recombinant, with either capital A and lowercase b, or lowercase a and capital B alleles. Thus, crossing over allows for the reassortment of linked genes.
So, from this video, we got to know that crossing over allows the reassortment of linked gene, and crossing over is the reason for the recombination and variation. To find out the answer to such question, Thomas Hunt Morgan and his colleagues tried to verify the chromosomal theory of inheritance through various experiments. Morgan chose the tiny fruit fly, that is Drosophila melanogaster, for his research. There was several reasons why he chose this tiny organism, that is Drosophila melanogaster. So now let's see what are the reasons for Morgan choosing this melanogaster for his experiment. Number 1, Drosophila melanogaster can easily grow in a synthetic medium in a laboratory. Morgan also found that a single mating of this organism produces a large number of offsprings. Moreover, the male and female is easily distinguishable. That means they are sexually distinguishable. That means we can easily recognize which one is the male Drosophila and which one is the female Drosophila. And another reason why he chose this Drosophila melanogaster because this tiny organism has 4 pair of chromosomes, making this analysis more easier for Morgan. Next, the reason Morgan why chose Drosophila melanogaster is these flies show several variations which could easily seen through low power microscope, so it helped Morgan to study their several generations. Finally, these flies have a life cycle of 2 weeks, which helped Morgan to study about their several generations within a short lifespan or within a short period of time. Here you can see they complete their life cycle in this stage, that is egg, first instar, second instar, third instar, pupa, and giving rise to adult. And this whole life cycle completes within a 2 week. That's why Morgan chose this organism so that he could study its several generations. So, finally, Morgan carried out a several dihybrid cross experiment with this Drosophila to study sex-linked genes. Let's analyze his experiments and the result in this next slide. Here you can see there is 2 different crosses were made, which Morgan did on the Drosophila melanogaster. First focus on this cross 1 or cross A. He started with the parental combination, that is yellow-white and wild type. Drosophila had same chromosome number as the same, his chromosome is represented as the same as in case of man, that is their male is represented by XY and their females are represented as XX, just like humans. In the first cross, Morgan took 2 different traits, that is body color and eye color. Body color and eye color. He chose these 2 traits. So, the body color in Drosophila was yellow body and brown body. Yellow body is represented as Y and brown body is represented as Y plus. Similarly, the eye color, that is he saw the eye color, that is white and red. The white is represented as W and red is represented as W plus. So in this first cross, observe this yellow-white female is crossed with a wild type male and the F1 generation is obtained as wild type female and yellow-white male. So, in the first generation, we see some variation. So, Morgan, when he further raised the F2 generation, he got the 4 males as wild type, white, yellow-white and yellow, and the 4 females he got in F2 generation, that is wild type, white, yellow-white and yellow. And he saw that in the cross 1 or cross A, the recombination rate is 1.3%. That is 1.3% in the first case. Now let's focus on this cross B. Here he considered the 2 characters, that is the eye color and body. So in the cross B he considered eye color and the wing size, That is, here you can see miniature. So he crossed a white miniature female with a wild type male and got a wild type female and a wild miniature male. And when he raised the F2 generation, he got 4 males as wild type, miniature, white miniature and white, and the 4 females, that is wild type, miniature, white miniature and white. After performing this cross B, he found the recombination rate is 36.4%. So, now observing these two cross, can you tell in which case the linkage is less in between the two genes? Is it in cross A or is it in cross B? Of course, it is in cross B because the recombination rate is seen, that is 36.4%. So, as we observe in this cross A, the recombination rate is 1.3%. So, the linkage between the two genes is more. So, the gene, that is body color and eye color gene, is physically very close to each other in case of this cross, or they are very linked. So, they linked transfer to the next generation. Hence, they show less recombination, that is 1.3%. But in cross B, the recombination rate is more, so the linkage between the two genes is less, that is the eye color gene and the wing size gene is less. The linkage between these two genes is very less. Here, now try to observe this picture and try to tell what this picture is trying to tell us. Well, you can see this child has red colored hair and the light colored skin, isn't it? That is, the gene for hair color and the gene for skin color are placed very close to each other. So, if the child has light colored skin, then it will automatically have the red colored hair, as the gene for hair color and the gene for skin color are placed very close to each other. That means they are linked. Students, if you have understood up to this, if you have understood the concepts up to this, what is taught, then try to answer these questions. Question number 1. Likelihood of two genes separating during crossing over depends on dash. So, likelihood of two genes separating during crossing over depends on the distance between two genes. Next question. Crossing over results in dash. Well, crossing over results in genetic variation, or we can say recombination. The cross that was performed by M.H. Morgan, Thomas Hunt Morgan, you saw some genes are only restricted to the X chromosome. In the Morgan's cross, we saw that some genes are only present on the X chromosome, not on the Y. So, what is the reason behind that? So, let's find out the answer in the next video.
While observing fruit flies, Morgan discovered a male that had white eyes instead of the typical red ones. He mated the white-eyed male with a homozygous red-eyed female. The entire F1 generation, whether male or female, had the dominant red eyes, just as Mendel would have predicted. However, the F2 generation held a surprise. Just as Mendel would have predicted, 3/4 of the offspring had red eyes, and 1/4 had white eyes. But there was a catch. Only males had white eyes. 1/2 of the males had red eyes, and the other half white eyes. If sex and eye color were being inherited independently of one another, as Mendel would have predicted, then 1/2 the white-eyed individuals should be female. Obviously, eye color was somehow linked to sex. In order to explain his results, Morgan hypothesized that while there is a gene for eye color located on the X chromosome, there is no corresponding gene on the Y chromosome. Thus, females, which receive 2 X chromosomes, also receive 2 genes for eye color, while males, which receive 1 X and 1 Y chromosome, receive only 1 gene for eye color. Looking at Punnett squares based on Morgan's hypothesis helps explain the results of his experiment. Morgan started with a white-eyed male that produced only 2 types of sperm. Sperm containing the X chromosome, and along with it, the alleles for white eyes, and Y chromosome sperm that contained no alleles for eye color. The female fruit fly, being homozygous for red eyes, produced only eggs carrying the alleles for red eyes. In the F1 generation, the female offspring are created when the X chromosome white eye sperm unite with the X chromosome red eye eggs. Because all the females inherit 1 white-eye gene and 1 red-eye gene, they are all heterozygous for eye color. The F1 generation males are created when Y chromosome no eye color gene sperm unite with X chromosome red-eye eggs. Thus, all males have red eyes as a result of the single eye color gene they receive via the X chromosome of their female parent. Crossing the males and females of the F1 generation produces an F2 generation in which 1/2 of the females are homozygous for red eyes, inheriting 1 red-eye gene from their mother's X chromosome and the other from their father's X chromosome, and 1/2 are heterozygous, inheriting the white-eye gene from their mother and the red-eye gene from their father. 1/2 of the males are red-eyed, inheriting a red-eye gene from the mother's X chromosome and no eye color gene from their father's Y chromosome. The other 1/2 are white-eyed, inheriting the white-eye gene from their mother's X chromosome and no eye color gene from their father's Y chromosome. The outcome of Morgan's Punnett square is consistent with the actual breeding outcomes. The Morgan hypothesis isn't just about predicting eye color in fruit flies. It is also relevant in explaining the inheritance of a number of genetic diseases in humans. Genes that are located on one sex chromosome but not the other are referred to as sex-linked. Nearly all sex-linked genes are found on the X chromosome and are often referred to as X-linked genes. Humans carry genes related to color blindness, hemophilia, and muscular dystrophy exclusively on the X chromosome. The fact that males have only 1 copy of the genes for these traits puts them at a much greater risk for these illnesses than females, as they have no chance of inheriting a second good gene that will mask the expression of a bad gene they may inherit.
So, this is all about linkage and recombination. If you have understood the concepts completely, now try to answer these questions. What is linkage? Linkage is a physical association between 2 genes. How crossing over leads to genetic diversity? Question number 2. Try to answer. Well, when crossing over occurs, it causes the exchange of non-sister chromatids of homologous chromosome, which results in variation or recombination, which leads to genetic diversity. Now try to answer this very short answer type. Question number 1. Crossing over takes place in which stage? Well, crossing over takes place in the Pachytene stage of prophase 1 of meiosis 1. Question number 2. Percentage of crossing over is more when dash? Well, percentage of crossing over is more when two genes are placed very far from each other, that is, they are present distantly. So, students, in today's session, we studied about linkage and recombination, how crossing over leads to recombination and recombination leads to genetic diversity. So, this is all about linkage and recombination and Thomas Hunt Morgan's experiment which he conducted on Drosophila melanogaster. Thank you.